Hello. Say we're going to take a quick peek at this study guide together here. DNA, structure and replication. How is genetic information stored and copied? Why? Deoxyribose, or DNA, is the molecule of heredity. It contains the genetic blueprint for life. For organisms to grow and repair damaged cells, each cell must be capable of accurately copying itself. So how does the structure of DNA allow it to copy itself so accurately? So if you look here in Model 1, the goal of this study guide is just to say, let's take a peek at some of the concepts of the structure of DNA and what we think it looks like. First off, the nucleotide is the building block of DNA, and it's right here. It's the monomer that helps build the polymer. And there are actually 4 different monomers, and they're different because they have different nitrogen bases, and we've been practicing that. And we know that adenine goes with thymine and guanine goes with cytosine. And here's the ladder model of DNA, and this is the model that we've been using this week. We could put a twist in it to show that it's the helix model, and this is more closely representing what DNA looks like, but for our simplicity, we're going to look at it this way. All right. So we just got a series of questions that you answer using the information up above. Very easy, very easy. Very easy. Okay. So let's look at question 4 here real quickly. And I think they've got a key by it because I think it's important. Refer to Model 1. When one nucleotide contains adenine, what type of base is adenine attached to on the opposite strand? That's just practicing Chargaff's rule. Very easy. Number 6 is also a key question. When one nucleotide contains cytosine, what type of base is the cytosine attached to on the opposite nucleotide strand? Again, practicing Chargaff's rules. This is going to be a real simple assignment, guys. Now we're going to talk about Erwin Chargaff, an Austrian-American biochemist who investigated the ratio of nucleotide bases found in DNA from a variety of organisms. From his research, as well as research by Rosalind Franklin and Maurice Wilkins, Watson and Crick developed the complementary base pair rule during their race to discover the structure of DNA. The complementary base pair rule states that adenine and thymine form pairs across 2 strands, and guanine and cytosine form pairs across 2 strands. Fill in the complementary bases on the strands below according to the base pair ruling. So they just want you to write underneath here what pairs up with A, what pairs up with T, C, A, and G. All right, go ahead and answer the rest of the questions there. Now, this picture actually talks about DNA replication, Model 2. And this is what you were doing in your activity the other day. You're showing a DNA helicase moving along and opening, and then we're showing free nucleotides getting attached to the open strand. And we just show here some information or questions looking at that information. This type of replication is called semi-conservative replication. Consider the meaning of the word semi, half. Conservative means to keep. So why do we have this phrase, semi-conservative replication? Why is DNA considered semi-conservative replication? It has to do with something of semi, half, and conserve, to keep. This is just a little diagram that shows us some of the information that Chargaff discovered as he was looking at it. When he looked at this, he said, okay, I'm going to look at cells from a human. And when I look at it from a particular human, I'm getting 31% adenine and 19% cytosine. So if I have 31 adenine and 19% cytosine, how much guanine, what percent should be guanine and what percent should be thymine? And then across this way, if you add those 4 numbers up,